In this video, we're going to look at pathogenicity. So to begin with, let's talk about three terms that you need to know. The first is infection. So microbes that are able to penetrate into a host, get past that host defense mechanism, and begin to replicate in the host, that's known as an infection. Once the microbe's able to get in a host and start to replicate. Now, it's a pathogen when during that infection, that microbe is able to do harm to the host and cause disease. And then virulence refers to what is the ability or the degree for which a microbe is able to cause a disease in another organism. So let's talk a little bit about pathogenicity. And remember, that's the ability of the microbe, and this can be a bacteria, a fungus, a virus, whatever. What's its ability to cause a disease in a host organism? Remember, not all microbes are pathogenic. In fact, many are harmless or even beneficial. So pathogenic microbes have developed various mechanisms to successfully infect and then multiply within the host, leading to the development of diseases. So the process of becoming pathogenic generally involves a series of steps, and we have it down to 5 steps, but it's important to note that not all pathogens follow these steps in the exact order for which I'm going to present them. Different pathogens may employ different strategies, but our 5 steps are portal of entry, attachment, evasion of host defense mechanisms, causing of disease or doing the damage, and then exiting the host. So to begin with, let's start with the first step, which is portal of entry. Pathogenicity begins with the selection of the appropriate portal of entry into the host, and that appropriate portal is known as the preferred portal. In human, it's going to be one of 3 things. It's going to be a mucous membrane, the skin, or the placenta. And the choice of the entry point is critical for that success for that pathogen. When looking at portal of entry, there's 2 other things that we need to consider. One is, where does the microbe originate? Does it come from another person or from our environment? If so, we call it exogenous. Sometimes the microbe might originate from inside the host as part of its normal biota, especially if a host gets sick or something along that time becomes compromised. In those cases, we refer to it as an endogenous microbe. So the portal of entries are going to be like the skin. How does the microbe get in through the skin? Because somehow that skin gets broken through a nick or abrasion or a bite, might come in through the conjunctiva, and sometimes they just burrow in under an intact skin. The gastrointestinal tract can be the mucous membranes. There can be a portal of entry, and that's going to come because of eating or drinking contaminated foods or water, or putting inanimate objects like pens, pacifiers, things like that that might have a microbe on it into our mouth, and then that can serve as a portal of entry. The respiratory tract, the mucous membranes there from breathing in respiratory droplets can be a portal of entry. And then, of course, the urogenital tract with the genital area and the urethra may also be portals of entry. Now, not only is it getting to the preferred portal of entry, but another consideration in step 1 is how many microbes are coming in at the same time. Because if you don't have enough microbes, depending on the pathogen itself or the body's defense mechanisms, a few microbes may not be able to get established and may not be able to actually go down that path to become full pathogenic state. So it's the number that also comes in. And there's 2 numbers that we refer to. One is LD50 and one is ID50. And this is based on clinical trials. And so the lethal dose 50 is the number of microbes required to kill 50% of the challenged animals, whereas the infectious dose 50 is the number of microbes that have to enter in order to cause disease in 50% of the challenged animals. So in step 1, to recap, the microbe has to enter the body through a preferred portal, and it has to have enough microbes enter in order to be able to progress all the way through the pathogenic stages. So once the microbe gets in, then the pathogens have to attach to or enter into those host tissues and establish a presence. And so for that to happen, it's going to often require adhesion factors or specialized structures to facilitate the microbe attaching to the host cells or to the host tissues. For instance, they may use specific structural proteins like fibronectin or fimbriae. And so, like, for instance, E. coli uses fimbriae. And remember, that's one of the gram-negative bacteria. So different structural components which will help that bacteria attach to the host in order to adhere and be able to establish itself. So step 3 is the step that it has to evade host defense mechanisms. So during step 1 and in step 2 here, as the microbes enter the body and as they are attaching to the host tissues, into the host cells, these mechanisms that the microbes use to get in and to become established actually alert the host that, hey, there's an invasion. So during these steps, the host is beginning to mount up an immune response. And generally speaking, that first immune response is going to be some cellular action through cells known as phagocytes, which can go out and engulf or eat other cells. Think of it, if you will, if you're not sure what they are, like kind of like how Pac-Man could go in and eat. So it's going to eat and bring those cells in and destroy them, or through chemical reactions. And there's others as well, but these are the two main ones. So our microbes, in order to be able to become pathogens, are going to have to be able to fight past the host defense mechanisms and survive those. So they're going to have to use chemicals and enzymes and cell structures and even cell invasion to be able to get past those host defense mechanisms. So let's talk about a couple of the microbe defense mechanisms. In order to get past those phagocytic cells, there's 3 different commonly used options that microbes will have. One is capsules. Some of our bacteria and others will have a slimy layer that goes over them, and that slimy layer makes it so that a phagocyte is not able to get a hold of that microbe and bring it in, and so it's not able to ingest it and to kill it and deactivate it. Some use structural components such as M proteins, and those M proteins then actually prevent the phagocytes from actually being able to adhere to it and to again bring that microbe inside. And then other microbes use a whole different tactic, and in this tactic they allow the phagocyte to engulf them, and then once they get inside that enemy cell, they release enzymes or chemicals that are going to then be dispersed and kill that cell that has engulfed it. And once the cell dies, then the microbe is released again and able to continue its pathogenic process. Another process that's used is what we call cell invasion. So sometimes in order to hide from the host defense mechanisms, the microbe is going to go in and actually insert itself into a host cell like a virus does. And then that way, by being hidden inside of the host cells, when this host is putting out a survey to see where is this foreign object or where's this foreign virus or cell or whatever it might be, it's not going to be able to see it because that microbe has hidden inside of the cell of the host itself. So just some different mechanisms, and as we go through chapters or units 5 and 6, we'll learn more different mechanisms. So after we get through with evading the host defenses, then we come to causing the damage, which is step 4. So when we talk about damaging the host, ultimately, remember the definition. In order to be a pathogen, the microbe has to do damage to the host. So the manifestation of the disease symptoms can range from all different kinds of things, but a lot of those are going to come from how the microbe caused the disease. So there are 3 main ways for which microbes can cause disease: from the production of toxins, through the production of enzymes, and they can do it indirectly by causing the host to mount an immune response that is so great that it overwhelms the host themselves, such as like what would happen in an anaphylactic shock scenario, and in that case, or an autoimmune scenario. In those cases, then the host is damaging itself. So let's look particularly at the first 2, enzymes and toxins, and how do microbes use those in order to cause diseases. So the first one is enzymes, and we're going to talk about exoenzymes. So in this case, the enzymes are going to be secreted by the microbes, and then those enzymes are going to cause damage to the host cells or tissues, or they're going to somehow promote disease to occur because of the spread of that microbe. So examples of exoenzymes: mucinase. Now remember, these are enzymes, so they're all going to end in ASE. So mucinase sounds like mucus, and that's exactly what it's going to do. Is that enzyme, when released by the microbe, is going to digest that protective mucus coating, and that can lead to disease. Hyaluronidase, okay, this is an enzyme that's going to help break down hyaluronic acid, which helps to kind of hold tissues and cells together. So that can do damage, but it can also allow for the cells to get, the microbes to get deeper into the tissue and again, farther away from the host cell defense mechanisms. So it not only does damage, but it can also allow that microbe to get deeper into the host tissues. Coagulase. This is one when released, it helps to coagulate plasma and blood, which can obviously lead to some significant host damage. And collagenase, that is going to digest collagen factors, which is again a structural component used in animal tissues. So you break it down, it's going to again allow that disease to spread throughout the body because it's going to allow that microbe to go deeper and deeper into the tissues. So again, enzymes can do direct harm, or they can just help break down those barriers so that the microbe can spread to other areas or get deeper into the tissues of the host. Toxins are also used from the microbes. And toxins, those are going to be specific chemical products, okay, that can actually act poisonous to other organisms, if you will. Now, the toxins are named for their target. So like if it's a neurotoxin, it's going to act on the nervous system. If it's a hemotoxin, it's going to lyse red blood cells, etc. There are two classifications of bacterial toxins. We have exotoxins and endotoxins. Now, this is just a general overview picture before we begin into a little bit of the specifics. First here, I want you to notice this is an exotoxin. These are the bacterial cells, because this is what we're referring to when these toxins are bacteria. Okay, so this is a bacterial cell, and these two are bacterial cells. Notice in exotoxins that the toxin is inside a cell, the living cell excretes it, and then it impacts tissues. In the endotoxin, notice that you don't see toxins inside. Why? Because the toxins are actually going to be part of the cell outer membrane here, and then the cell dies and releases the toxin. So now let's look a little bit closer. So let's start with exotoxins. Exotoxins, again, are produced within the bacteria, and they're released for transport out of the host. So made inside, transported out. They're going to be protein in nature, water soluble. They're going to be tissue specific. That is very important to recognize, that they're going to be tissue specific, and they're going to speed up specific reactions within the host cells. Okay, now I don't know if it's going to be an existing one or if it's going to initiate another. It depends on that toxin. All right, but they are going to start or speed up reactions within the host. Now, as a general rule here, it does say that they're produced by gram-positive, and that is correct, but it doesn't mean that it's only gram-positive. It's just majority of the toxin-producing bacteria are gram-positive. Now, although there's various kinds of exotoxin, the most common type is what we call an AB exotoxin. And we call it an AB exotoxin because there's two subunits to the toxin. One is A, and that's the catalytic unit, the one that's going to do the change, and then B, which is the one that is going to be the binding toxin. So what does that mean? The bacteria produce this AB toxin. Now remember, exotoxins are tissue specific, and it's tissue specific because of the binding unit. So it gets released by the bacteria. The binding unit, B, will attach to a very specific host cell because of that binding unit finding a particular protein or substance on that specific target cell wall. It's going to, or cell membrane, it's going to target, it's going to adhere. Then that adhering causes the host cell to take that toxin inside. So it endocytoses and brings it inside, and then once it gets inside, there's a separation between the A and the B units. And then the A unit, which is the active or the catalytic unit, is going to then go inside the host cell, and again, it's either going to increase a current reaction that's occurring, or it's going to start a reaction that hasn't been occurring. But that is going to be the AB toxin. So they're going to get inside their host cell, and they're going to initiate a particular change within that metabolic process of the cell, and that change that they initiate is going to cause damage to that cell or to the host overall. So again, the exotoxins are going to be named for that system for which they're going to be impacting the most. For instance, the cytotoxins, they're going to kill host cells or inhibit their function. Neurotoxins are going to be the exotoxins which are going to interfere somehow with nerve transmissions or transports. And then the enterotoxins will be one that affect the epithelial tracts or the epithelial cells of the GI tract. Now, because of the specificity and because of the way this one works, a couple things that I want to point out. First of all, you should notice that the cell, the bacterial cell, is secreting the toxin. The cell itself does not do harm to the host for which it has invaded. It's the toxin that does the damage. However, you can go in and kill the bacterium, but if it has already released thousands of toxic molecules, it's still going to be doing damage. So when the body has to clear out or defend against a bacteria that's producing a toxin, it's going to have to do two things. It's going to have to get rid of the bacteria, but it's also going to have to clear out the toxin. And what we use to clear out a toxin is what's known as an antitoxin, and that will neutralize out the toxin. So with exotoxin, the takeaways are to remember that it is tissue specific. It is going to start or amplify specific reactions in the host, which are going to lead to damage. That it is going to be one that is mostly by gram-positive, and you've got to get rid of both the bacterial cell and the toxin in order to alleviate the host from the symptoms. Now let's talk endotoxins. Endotoxins are quite different than exotoxins. Endotoxins are structural components of the bacterial cell, and they're actually going to be found as part of that outer membrane. You might remember in chapter 2 when we were doing the structure of a bacteria, we talked about gram-negative bacteria having an outer membrane, and embedded in that outer membrane was a chemical known as lipopolysaccharide, or LPS. So in the case of endotoxins, what is going to happen is that the cell is going to, let me go back to that picture, the cell is going to die or rupture, and when it does, it breaks into small components, and those components from that outer cell membrane are going to be released, and the LPS toxin gets released from the dead or dying cell. When it gets released, then it's going to go systemically into the tissues or into the fluids of the body, and it's going to cause just a generalized effect. And that means it's going to have things such as fever, inflammation, chills, weakness, okay? And if a lot of it gets released, so there's a large infection of these gram-negative bacteria and a large number are dying off, then it can cause the body to go into shock because of a massive outpouring of LPS into the host system. So endotoxin is for gram-negative, and it is a release of a component embedded within that outer membrane. So just kind of a quick review of some of the defense mechanisms, and this is also how the cell can, it's basically step 3 and step 4 as mentioned. These can be overlapping at times. So these could be defense mechanisms against the host defenses, or it can be ways too that it's causing damage. So this is just looking at E. coli. So there's different enzymes that E. coli secretes that can help it to evade host defenses, and those enzymes actually also do damage and cause diseases. The flagella, that can help it to evade, but it can also help it to get deeper into the host tissues to do more damage. Again, it's going to have those exotoxins in there, and it's going to have that LPS because it is a gram-negative. So exotoxins, endotoxins, enzymes, and various chemicals. E. coli uses them all in methods to evade the host defenses and to do cell damage. So we're now to the last step, and that's exiting the host. So remember, the goal of the microbe that is pathogenic is to cause diseases and to do a large amount of causing diseases. So if it gets into a host and it causes disease there, but then it can't spread, then as the host dies, it too will die. So one of the factors that you have to consider is that the last step is to exit the host. So how does the microbe exit a host? It's got different ways to get in, it's got different ways to get out. So ways that it can leave or exit the host, different mechanisms. So the host who has the bacterial infection or the viral infection or whatever may be going on might be able to sneeze or cough and do respiratory droplets, and then the microbe can get out and spread to others. It might be through needles, from either from having blood drawn or from sharing of needles. It might be from insect bites. It might be from urine and feces and getting fecal contaminated into the waters or to foods, etc. Or it might just be from lesions or wounds that it's able to spread in that method as well. So again, Not all microbes are going to follow the exact steps in the order that we just went through. And as you could tell, some of those kind of overlap. So there's not necessarily a definite start and end to each step. But there are 5 pieces to this pathogenic disease state. Okay, the first is that the microbe does need to enter into the host. It needs to use a preferred portal, and it needs to have enough microbes enter in order for it to be able to get all through the 5 stages. Once it gets inside the host, it's going to have to be able to attach itself and to establish itself so that it can then put up a defense and evade those host defense mechanisms, be it cellular, chemical, etc. So step 3 is evading those host defense mechanisms. And then in order for it to actually be pathogenic and not just cause an infection, but to be pathogenic, it's got to do damage and cause, by causing that damage, that means it's a disease causer. And so it's going to do damage through toxins or enzymes and scenarios such as that. And then finally, the last step is that it's going to have to be able to spread from one host to another. So it's going to have to be able to exit its host and go to a new host. And for how it goes from one host, the transmission to a next, is the next video. So watch that spread of infection video next.